The African savanna provided the environment for early hominid evolution. Volcanic features dotted the landscape, indicating past geological activity. The savanna's diverse ecosystem supported the development of early human ancestors. The open environment presented both opportunities and challenges for survival. Early hominids adapted to the savanna through bipedalism and tool use. A volcanic eruption begins with the violent expulsion of molten rock, ash, and gases from the Earth's interior. Volcanic ash, composed of fine particles of pulverized rock, rapidly blankets the surrounding landscape. The layer of ash creates a soft, impressionable surface ideal for preserving footprints. Over time, the ash hardens and solidifies, permanently preserving the footprints as a record of past life. Volcanic ash settles, forming a layer on the ground. The ash layer consists of fine, particulate material. The ash layer becomes soft and damp, ideal for preserving footprints. The damp ash layer provides a high-resolution record of surface impressions. Animals traverse the ash-covered savanna, leaving footprints as evidence of their passage. Footprints in the ash serve as a temporary record of the animal's movements across the landscape. The ash layer provides a stark contrast, highlighting the impact of the animals on the environment. Different animal species leave distinct footprints, revealing the diversity of wildlife in the area. The footprints fade over time, illustrating the ephemeral nature of traces in the environment. Fresh footprints are initially imprinted in soft volcanic ash. Subsequent rainfall saturates the ash layer. Water percolates through the ash, dissolving minerals. Dissolved minerals precipitate, binding ash particles together. The hardened ash layer preserves the footprints. Over millions of years, the footprints remain as fossilized records. The Laetoli site in Tanzania is the location where the hominin footprints were discovered. This site provides critical evidence of early hominin bipedalism. The footprints offer a direct record of hominin locomotion from millions of years ago. The Laetoli footprints are a significant paleontological find, providing insights into early human ancestors. These footprints contribute to our understanding of the evolution of upright walking in hominins. Mary Leakey's expedition involved meticulous excavation of significant archaeological sites. The team's work focused on uncovering hominid fossils and ancient tools. Systematic layering and documentation were crucial to understanding the site's history. Careful analysis of artifacts provided insights into early human behavior and technology. The expedition's findings contributed significantly to the understanding of human evolution. Reconstructed footage depicts the challenges and dedication of archaeological fieldwork. The process of uncovering fossilized footprints requires meticulous and delicate work. Careful removal of sediment layers is essential to reveal the footprints without causing damage. Specialized tools are used to gently clear away debris and expose the fossilized surface. Documentation through photography and 3D scanning captures the precise details of each footprint. The unearthing process aims to preserve the integrity and context of the fossilized footprints for scientific study. The footprint trail indicates the path taken, showing direction and distance traveled. Trail length is visually represented to quantify the extent of the journey. The direction of the footprint trail reveals the route followed. The overview provides a comprehensive understanding of the path's trajectory. Footprint morphology involves analyzing the shape and depth of footprints. Comparative analysis of footprint morphology aids in differentiating between human and ape footprints. Shape and depth are key morphological characteristics used in footprint analysis. Variations in footprint morphology provide insights into gait, weight distribution, and species identification. The Laetoli footprints provide direct evidence of early human bipedalism. Ape footprints typically show a divergent big toe and a less pronounced arch compared to human footprints. Modern human footprints exhibit a distinct arch and an aligned big toe, indicative of efficient bipedal locomotion. Laetoli footprints share key characteristics with modern human footprints, suggesting similar biomechanics. Comparative analysis reveals evolutionary adaptations in foot structure related to bipedal walking. Fossilized footprints provide direct evidence of bipedal locomotion in early hominids. A rounded heel is a key anatomical feature, indicating weight-bearing during upright walking. The presence of an arched foot structure further supports the ability to walk efficiently on two legs. These footprints demonstrate the biomechanical adaptations necessary for sustained bipedalism. Gait analysis involves the systematic study of animal locomotion, particularly bipedal walking. Hominid gait is characterized by upright posture and bipedal locomotion. Key elements of gait include stride length, cadence, and foot pressure distribution. Animation can effectively demonstrate the biomechanics of hominid walking. Upright walking provides advantages such as increased visibility and freeing the hands for tool use. Australopithecus afarensis is a species of early hominin believed to be capable of bipedal locomotion. Reconstruction efforts focus on accurately representing skeletal structure and musculature based on fossil evidence. The species' physical characteristics suggest a transitional stage between apes and modern humans. Footprints discovered in Laetoli provide direct evidence of bipedal walking in Australopithecus afarensis. Visual reconstructions aid in understanding the morphology and behavior of early hominids. Australopithecus afarensis exhibits skeletal adaptations indicative of bipedal locomotion. The pelvis of Australopithecus afarensis is shorter and broader compared to that of modern humans, aiding in upright posture. The femur of Australopithecus afarensis exhibits a distinct angle from the hip to the knee, known as the bicondylar angle, facilitating efficient bipedal walking. The spinal curvature in Australopithecus afarensis is less pronounced than in modern humans, reflecting a transitional stage in bipedal adaptation. The foot of Australopithecus afarensis retains some arboreal features, such as slightly curved toes, alongside adaptations for bipedalism. These skeletal differences reflect the evolutionary transition from arboreal to terrestrial locomotion in early hominins. Bipedalism frees the hands, enabling the carrying and manipulation of objects. An upright posture enhances the field of vision, allowing for better observation of the surrounding environment. Improved vision from bipedalism aids in spotting predators and resources from a distance. Carrying objects while walking upright facilitates migration and resource transport. The combination of free hands and enhanced vision provides a significant survival advantage. The ability to carry objects with free hands provided early hominids with significant advantages. Free hands enabled the transportation of resources over longer distances. Carrying tools facilitated more efficient hunting and gathering practices. The transport of food allowed for resource sharing within social groups. Free hands also contributed to improved defense capabilities. The development of bipedalism was crucial for freeing the hands and advancing tool use. Standing upright enabled early hominids to scan the horizon more effectively. An elevated perspective allowed for earlier detection of potential threats. Increased visibility provided a survival advantage by facilitating predator avoidance. Upright posture also improved the ability to locate potential prey across long distances. The ability to see further enhanced hunting and gathering strategies. Early predator or prey detection contributed to increased survival rates. Footprint trails can be analyzed to infer social behavior among hominids. Analyzing the proximity and direction of footprints reveals whether hominids walk together as a group. Consistent spacing and parallel alignment suggest coordinated group movement. Variations in footprint size and depth can indicate age and weight differences within the group. Changes in gait and stride length may reflect different activities or social roles within the group. Footprint analysis provides insights into the social dynamics and interactions of early hominid communities. Australopithecus afarensis were early hominids who lived in Africa millions of years ago. Fossilized footprints provide direct evidence of their bipedal locomotion and social behavior. Reconstructions are based on skeletal remains and footprint patterns. These hominids exhibited a mix of ape-like and human-like characteristics. The group dynamic suggests cooperative behavior and social structure. Footprint size variation can be analyzed to estimate the height of individuals. Larger footprints generally correlate with taller individuals, while smaller footprints suggest shorter individuals. The length and width of a footprint are key parameters in estimating an individual's size. Statistical models and regression analysis can be applied to footprint measurements to refine height estimations. Variations in footprint size can also provide insights into an individual's weight and build. Footprint analysis suggests the possibility of a family group, including adults and juveniles. Variations in footprint size and depth can indicate different age groups within the group. Spacing and direction of footprints may reveal movement patterns and social dynamics. The presence of both adult and juvenile footprints supports the hypothesis of a family unit traveling together. Further analysis, including gait and stride length, can provide additional insights into the group's composition. Footprint analysis provides insights into the size and spacing of hominid family members. The reconstruction of a hominid family can be inferred from fossilized footprint trails. Size variations in footprints suggest the presence of adults and children within the hominid family. Spacing between footprints reveals information about the gait and walking speed of each family member. Reconstructed hominid family walking together based on footprint size and spacing analysis. The arrangement of footprints provides clues about the social dynamics and relationships within the family. The Laetoli footprints provide direct evidence of bipedalism in early hominids. Australopithecus afarensis, the species believed to have made the Laetoli footprints, existed approximately 3.6 million years ago. Early hominid evolution is characterized by the gradual development of bipedal locomotion. The timeline of hominid evolution spans millions of years, with various species emerging and diverging. Australopithecus afarensis is positioned within the early stages of hominid evolution, preceding later Homo species. Fossil evidence, including footprints and skeletal remains, provides crucial insights into hominid evolution. An evolutionary tree illustrates the relationships between different hominid species. Australopithecus afarensis is a significant early hominid species known for its bipedalism. Bipedalism, or walking on two legs, is a key evolutionary adaptation in hominids. The position of Australopithecus afarensis on the tree indicates its ancestral role in human evolution. Fossil evidence supports the placement of Australopithecus afarensis in the hominid evolutionary tree. Evolutionary trees are constantly updated as new fossil discoveries and genetic data emerge. The Laetoli footprints provide crucial evidence for understanding early human bipedalism. These footprints confirm that early hominids walked upright approximately 3.6 million years ago. Analysis of the footprints reveals insights into the gait and weight distribution of early hominids. The Laetoli footprints are a significant discovery for tracing the evolutionary path of humankind. The Laetoli footprints provide direct evidence of early hominin bipedalism. These footprints, discovered in Tanzania, date back approximately 3.6 million years. Comparison of the Laetoli footprints with skeletal remains, such as Lucy, provides a more complete understanding of early hominin anatomy and behavior. The Laetoli footprints and Lucy are crucial pieces in the puzzle of human evolution, offering insights into locomotion and morphology. Australopithecus afarensis exhibits a mosaic of ape-like and human-like skeletal features. Compared to modern humans, Australopithecus afarensis had a smaller cranial capacity, indicating a smaller brain size. Australopithecus afarensis possessed longer arms relative to legs, suggesting arboreal adaptations. The pelvis of Australopithecus afarensis exhibits adaptations for bipedalism, though less efficient than that of modern humans. Dental arcades in Australopithecus afarensis are intermediate between the rectangular shape of apes and the parabolic shape of humans. Climate change is a significant environmental factor that may have influenced the evolution of bipedalism. Habitat shifts, such as the transition from dense forests to open savannas, could have favored bipedal locomotion. Bipedalism may have provided advantages in spotting predators and traversing open landscapes. The need to carry food and tools over longer distances in open environments may have also driven the selection for bipedalism. Millions of years ago, East Africa was characterized by dense, lush forests. Gradual climate changes led to a reduction in rainfall and increased aridity. As forests receded, grasslands began to expand across the landscape. The expansion of grasslands created new ecological niches and opportunities. Over time, East Africa transformed from a forested region to a predominantly grassland environment. The Laetoli site's geological context is defined by significant volcanic activity. Volcanic eruptions deposited layers of ash, which subsequently hardened into tuff. These tuff layers preserved the famous hominin footprints. Sedimentary layers above and below the tuff provide a chronological record of the site. The interplay of volcanic activity and sedimentation created ideal conditions for fossil preservation at Laetoli. The Laetoli site stratigraphy reveals layers of volcanic ash and sediment. Volcanic ash layers provide a timeline for dating the hominin footprints. Sediment layers preserve the hominin footprints discovered at Laetoli. Stratigraphy helps reconstruct the environmental conditions at the time the footprints were made. Radiometric dating of volcanic ash is a method used to determine the age of footprints. Radiometric dating measures the decay of radioactive isotopes within the volcanic ash. The ratio of parent to daughter isotopes indicates the time elapsed since the volcanic eruption. By dating the volcanic ash, the age of the footprints preserved within the ash layer can be estimated. Radiometric dating determines the age of materials by measuring the decay of radioactive isotopes. Radioactive isotopes decay at a constant, known rate, allowing precise age calculations. The ratio of parent isotopes to daughter isotopes indicates the time elapsed since the material formed. Different isotopes with varying half-lives are used to date materials of different ages. Carbon-14 dating is suitable for dating organic materials up to approximately 50 years old. Uranium lead dating is used for dating very old rocks, often billions of years old. Specific environmental conditions are crucial for the long-term preservation of footprints. Rapid burial by sediment protects footprints from erosion and weathering. The composition of the sediment affects the preservation quality of the footprint. Arid climates and the absence of water help prevent the breakdown of footprint-bearing materials. Mineralization can further enhance footprint preservation by replacing organic material with durable minerals. Microscopic analysis reveals the detailed structure of the ash layer. Footprint impressions within the ash are discernible at a microscopic level. The ash layer's composition includes fine particulate matter and trace elements. High-resolution imaging allows for precise measurement of footprint depth and dimensions. Microscopic analysis aids in identifying the source and age of the ash deposit. The footprint's micro-details provide valuable insights into the object that created it. Footprint analysis involves examining specific details, such as toe impressions. The shape and depth of toe impressions can provide insights into gait and potential abnormalities. Arch support characteristics are crucial for determining foot type and biomechanics. Analysis of arch support helps in identifying individual walking patterns and potential orthopedic issues. Detailed footprint analysis aids in forensic investigations by providing unique identifying characteristics. The human foot is a complex structure comprised of 26 bones, 33 joints, and over 100 muscles, tendons, and ligaments. The foot is divided into 3 sections: the forefoot, midfoot, and hindfoot. The arches of the foot, including the medial longitudinal arch, lateral longitudinal arch, and transverse arch, provide support and flexibility. The plantar fascia, a thick band of tissue on the bottom of the foot, supports the arch and acts as a shock absorber. The ankle joint allows for dorsiflexion and plantar flexion, enabling the foot to move up and down. These anatomical features allow the human foot to efficiently support the body's weight and facilitate bipedal locomotion. The gait cycle is a repetitive pattern of movements that occurs during walking. The cycle begins with heel strike and ends when the same foot contacts the ground again. The gait cycle is divided into 2 main phases: stance phase and swing phase. Each phase involves a sequence of events, including heel strike, foot flat, midstance, toe off, and swing through. The human gait cycle is composed of distinct phases, each with specific biomechanical functions. The stance phase begins with initial contact, where the heel strikes the ground. During mid-stance, the body's weight is supported by a single leg. The terminal stance phase involves heel lift and weight transfer to the forefoot. The swing phase includes initial swing, mid-swing, and terminal swing, preparing the leg for the next stance phase. Bipedalism and quadrupedalism exhibit different energy efficiencies during locomotion. Bipedalism can be more energy efficient at slower speeds due to pendulum-like energy exchange. Quadrupedalism tends to be more energy efficient at higher speeds due to increased stability and propulsive force. Energy efficiency in both gaits depends on factors such as body mass, limb length, and terrain. Energy expenditure differs significantly between bipedal and quadrupedal locomotion. Bipedal walking involves a unique balance and gait cycle, influencing energy use. Quadrupedal locomotion utilizes a more stable stance, generally resulting in lower energy expenditure per distance. Comparative analysis reveals variations in metabolic cost between two-legged and four-legged movement. Factors such as body mass, speed, and terrain affect energy expenditure in both bipedal and quadrupedal movement. Bipedalism provided early hominids with an enhanced ability to spot predators and prey across the tall grasses of the African savanna. Standing upright freed the hands, allowing for the transport of food, tools, and offspring over long distances. Bipedal locomotion is more energy efficient than quadrupedalism for long-distance travel in open environments. The elevated posture facilitated better thermoregulation by reducing the surface area exposed to direct sunlight and increasing exposure to cooling breezes. Bipedalism allowed for the use of tools and weapons, providing a significant advantage in hunting and defense. The savanna environment presented both challenges and opportunities for early hominids. Bipedalism provided hominids with an elevated vantage point for spotting predators and resources. Walking upright freed hands for carrying food, tools, and offspring across the savanna. Bipedal locomotion may have also aided in thermoregulation by reducing exposure to direct sunlight. Bipedalism, or walking upright on two legs, freed the hands for carrying and manipulating objects. This manual dexterity facilitated the development and refinement of tool use. Tool use, in turn, provided early hominids with advantages in hunting, gathering, and defense. The ability to create and utilize tools contributed to cognitive development and problem-solving skills. The coevolution of bipedalism and tool use represents a significant milestone in human evolution. Hominids utilized simple tools, such as stones, for basic tasks. Stones were employed to crack nuts, providing a valuable food source. Hominids also used tools for digging, enabling them to access roots and tubers. The use of tools provided hominids with a significant survival advantage. Bipedalism freed early hominid hands, enabling tool use. Tool use required and promoted increased cognitive abilities in hominids. The development of bipedalism and tool use created selective pressures favoring larger brain size. Increased brain size allowed for more complex social structures and communication. Tool use and social interaction further drove brain development through learning and innovation. The coevolution of bipedalism, tool use, and brain development led to the emergence of modern humans. Australopithecus afarensis exhibits a smaller brain size when compared to modern humans. Brain size comparison reveals evolutionary trends among different hominid species. The cranial capacity of Australopithecus afarensis is significantly less than that of Homo erectus. Increased brain size in hominids correlates with advancements in cognitive abilities and tool use. Brain size is an important but not sole indicator of intelligence and behavioral complexity. The Laetoli footprints provide direct evidence of bipedalism in early hominids. Footprint analysis reveals insights into gait, stride length, and weight distribution of early hominids. Brain size, inferred from fossil skulls, is a key indicator of cognitive capacity in hominid evolution. Tool use, evidenced by stone tools, suggests problem-solving abilities and manual dexterity in early hominids. Comparative analysis of fossil evidence helps reconstruct the cognitive evolution of early hominids. The development of cognitive abilities facilitated adaptation and survival in changing environments. Reconstruction of hominid behavior provides insights into early human social structures. Foraging for food was a fundamental aspect of hominid survival. Hominid diets likely consisted of fruits, roots, and small animals. Cooperation and communication were essential for successful foraging. Early hominid social interactions involved complex communication methods. Reconstructed behaviors offer clues about the evolution of human intelligence. Footprint evidence provides insights into the social structure of early hominids. Analysis of footprint size and spacing can indicate group composition and social dynamics. Anthropological data, combined with footprint analysis, helps infer social behaviors such as cooperation and migration. Inferred social structures range from small family units to larger, more complex social groups. Hominids' social interactions include activities such as grooming, which strengthens social bonds. Sharing food is a crucial social behavior among hominids, fostering cooperation and resource distribution. Social interactions among hominids promote group cohesion and survival. These interactions are fundamental to the development of complex social structures in early hominid communities. Early hominids adapted to changing environments through bipedalism, allowing for better visibility and freeing hands for tool use. The development and utilization of simple tools aided early hominids in acquiring food and defending against predators. Early hominids faced challenges such as limited food sources, exposure to predators, and fluctuating climates. Social structures and cooperative hunting strategies improved survival rates among early hominid groups. Brain size and cognitive abilities gradually increased, enabling more complex problem-solving and adaptation strategies. Hominids faced challenges in securing water sources in arid environments. Early hominids developed strategies to avoid predators in their natural habitats. Cooperation and tool use were crucial for hominids to overcome environmental obstacles. Adaptation to changing climates was essential for the survival of hominid populations. Climate change significantly altered environments, impacting early hominid habitats and food sources. The drying of African forests led to the expansion of grasslands, favoring hominids adapted to open environments. Changes in vegetation and climate forced hominids to adapt their diets, leading to evolutionary pressures. Bipedalism became advantageous in open environments, allowing hominids to see over tall grasses and travel efficiently. Climate variability may have driven the evolution of larger brains and increased cognitive flexibility in hominids. The development of tool use and social cooperation among hominids was influenced by environmental challenges. Millions of years ago, East Africa experienced a significantly wetter climate. Over time, the climate of East Africa gradually transitioned towards drier conditions. This shift resulted in the formation of vast savannas and arid landscapes. The changing climate impacted the distribution and evolution of plant and animal life. Geological records and fossil evidence support the transition from wetter to drier climates. Current climate models predict continued aridification in East Africa, posing challenges for water resources and agriculture. The loss of forests significantly altered the environment inhabited by early hominids. Expansion of grasslands replaced forested areas, creating new ecological niches. Hominids were compelled to adapt to these new open environments. Changes in habitat influenced hominid locomotion, favoring bipedalism. Altered diets became necessary as food resources shifted from forest fruits to grassland vegetation. Habitat loss and grassland expansion played a crucial role in driving hominid evolution. The transition from dense forests to open grasslands altered hominid habitats. Deforestation and climate change contributed to the expansion of grasslands. Hominids adapted to grasslands by developing new survival strategies. Bipedalism became more advantageous for traversing open terrain. Tool use evolved to include hunting weapons and processing of grassland resources. Social structures adapted to facilitate cooperative hunting and resource sharing. Evolutionary pressures, such as environmental changes, played a key role in the development of bipedalism in early hominids. Bipedalism provided advantages like improved visibility over tall grasses, aiding in predator detection and resource location. Freeing the hands allowed early hominids to carry food, tools, and infants, enhancing survival and reproductive success. Changes in climate and resource availability favored hominids who could efficiently travel longer distances in search of sustenance. The development of bipedalism and related adaptations represents a complex interplay of environmental factors and natural selection. Early hominids underwent significant evolutionary adaptations affecting skeletal structure. Changes in pelvic structure facilitated bipedalism, allowing for more efficient locomotion. Limb adaptations, including longer legs and modified feet, further enhanced bipedal movement. Evolutionary pressures also led to an increase in brain size among early hominids. Increased brain size correlates with enhanced cognitive abilities and tool use. Genetic evidence provides crucial support for fossil evidence in tracing early hominid evolution. Comparative genomics reveals genetic similarities and differences between modern humans and extinct hominid species. Analysis of ancient DNA extracted from hominid fossils confirms evolutionary relationships and divergence times. Mitochondrial DNA, mtDNA, analysis helps trace maternal lineages and population migrations of early hominids. The presence of specific gene variants associated with traits like brain size and bipedalism provides insights into hominid adaptation. Genome-wide association studies, GWAS, identify genetic markers linked to uniquely human traits, differentiating us from other primates. Phylogenetic trees illustrate the evolutionary relationships among different hominid species. Genetic analysis provides evidence for the shared ancestry and divergence points of hominid lineages. Modern humans share a common ancestor with other hominid species, but have evolved distinct genetic traits. The diagram illustrates the genetic connections and evolutionary timeline leading to modern humans. Early hominids originated in Africa and subsequently migrated to other continents. These migrations were driven by factors such as climate change, resource availability, and population growth. The routes taken by early hominids often followed coastlines and river valleys, providing access to water and food. Different hominid species migrated at different times, leading to a complex pattern of dispersal and adaptation. Hominids originated in Africa and began migrating to other continents. Early hominid migrations spread into Asia and Europe. Migration patterns were influenced by climate change and resource availability. Different hominid species adapted to diverse environments during their migrations. The Americas were populated via the Bering Land Bridge during glacial periods. Hominid migration led to the global distribution of humans and their diverse cultures. Cultural evolution plays a significant role in the development of human societies. Cultural evolution involves the transmission of knowledge, beliefs, and practices across generations. This transmission leads to cumulative cultural change over time. Cultural evolution enables societies to adapt to their environments and solve complex problems. Cultural evolution can result in diverse social structures, technologies, and belief systems. The process of cultural evolution is influenced by factors such as innovation, diffusion, and selection. Early humans developed tools for survival and improved living conditions. Cave paintings represent early forms of artistic expression and communication. Early cultural practices included the creation of ornamental objects and personal adornments. The development of communal living and social structures marked a significant step in early human culture. Technological advancements have enabled human adaptation to diverse environments. Controlled fire provided warmth, protection, and cooking capabilities, expanding habitable zones. The development of clothing and shelter offered protection from extreme weather conditions. Agriculture and irrigation systems enabled stable food supplies in previously uninhabitable areas. Advanced transportation technologies facilitate resource acquisition and migration across vast distances. The development of agriculture marked a pivotal moment in human innovation, enabling settled communities. Domestication of animals provided new sources of food, labor, and companionship, transforming human societies. These innovations led to increased food production and population growth, fostering further technological advancements. Agriculture and animal domestication represent foundational innovations that shaped the course of human civilization. Human societies have exhibited increasing social complexity over time. Social complexity is characterized by increased specialization and division of labor. Hierarchical structures and institutions emerge to manage complex social interactions and resource allocation. Technological advancements often drive increases in social complexity. Increased population density and urbanization are consequences of social complexity. Social complexity can lead to both increased efficiency and potential social inequalities. Hunter-gatherer bands represent one of the earliest forms of human social organization. These bands typically consist of small, kin-based groups with egalitarian social structures. Early agricultural villages marked a transition towards more settled and complex social systems. The shift to agriculture led to increased population density and the development of specialized labor roles. Human activities significantly impact the environment through various mechanisms. Deforestation leads to habitat loss, reduced biodiversity, and increased carbon dioxide levels. Industrial emissions contribute to air and water pollution, affecting human health and ecosystem stability. Agricultural practices, including pesticide use and intensive farming, can degrade soil quality and contaminate water sources. Overconsumption and waste generation result in resource depletion and pollution from landfills and improper disposal. Deforestation significantly reduces biodiversity and disrupts natural ecosystems. Industrial pollution releases harmful chemicals and toxins into the air and water. Human activities contribute to increased greenhouse gas emissions, accelerating climate change. Unsustainable resource consumption depletes natural reserves and endangers future generations. Sustainability emphasizes meeting present needs without compromising the ability of future generations to meet their own needs. Responsible stewardship of the planet involves managing resources and ecosystems to ensure long-term health and productivity. Environmental, social, and economic factors are integral components of sustainability. Sustainable practices aim to minimize environmental impact through reduced consumption, waste, and pollution. Adopting sustainable strategies promotes long-term resilience and reduces vulnerability to environmental and economic shocks. Renewable energy sources, such as solar and wind power, represent key sustainable practices. Conservation efforts, including forest preservation and water management, are crucial for environmental sustainability. Sustainable agriculture promotes environmentally friendly farming practices. Recycling programs and waste reduction initiatives minimize environmental impact. Sustainable transportation options, such as electric vehicles and public transit, reduce carbon emissions. The Laetoli site is an active research location, continually yielding insights into early hominin behavior. Preservation of the Laetoli footprints is crucial for future generations of scientists and researchers. Ongoing research at Laetoli employs advanced techniques to analyze and interpret the fossilized footprints. The Laetoli footprints provide direct evidence of bipedalism in early hominins. The unique environmental conditions at Laetoli contributed to the exceptional preservation of the hominin footprints. Continued study of the Laetoli site enhances understanding of human evolutionary history. Please subscribe to the channel and hit the notification bell so that you will be updated for more videos.